nutrient auger plate that you made a couple weeks ago, been sitting in the cooler waiting for you, all right? Practice your streaking technique for isolation. Everyone cool on that, or does that confuse you? All right, number 2, you will grab a mannitol salt auger plate, and you will streak for isolation on that plate also, just like you did on your personal nutrient auger plate. Is that clear?
Yes.
Okay. Thirdly, you'll grab an EMB plate, eosin methylene blue plate, and I'll explain this stuff to you in just a second, and you will streak for isolation on this auger plate. Make sure when you put your plates in the 37-degree centigrade incubator, I just had 37 since we had 2, 4, 37, 4. Okay, all right. 37-degree centigrade, you store it upside down, it's labeled properly, and you're going to be using a new mixture. Remember how we used a mixture a couple weeks ago? Yeah, I made up a new one. Okay, what's that mixture made up of? This is really quite a varied mixture. Bacillus subtilis. What's one of the unique properties of bacillus? Good. Awesome. Who said that?
We said it at the same time.
Okay, that's great, man. You guys are freaking awesome. That's great. Spore former. I've been punishing the mixture so the bacillus should form spores. So it shouldn't be a problem. But if some of those spores sporulate and populate, it's really hard to get isolation. Very hard, because bacillus likes to spread. The colonies like to spread out. And you'll see that. So if the bacillus forms, that's bad on me. Okay, I didn't punish it enough. I've been keeping it in the cooler, shutting it down so it would form spores. But we'll see. Okay. The second component is Staphylococcus aureus. What's one of the things that we think of with Staphylococcus aureus? It's a human pathogen, so it needs to be handled carefully. Okay. The third and final in this mixture is E. coli. What about E. coli?
Be careful.
Yeah, it's a human pathogen too. So don't be drinking the mixture. Okay? Where's the mixture? Where's the mixture that you each are going to use to make it?
37-degree cooler.
They're sitting on very good, so sharp, high five. Ooh. There you go. They're on top of the incubator. Okay? So you will each practice sterile technique, flaming the tube flaming the tube after the cap comes off. Make sure that your loop, you're using a loop to streak for isolation, okay? And transfer from the broth to the plate and streak. So that's all you're doing, is these 3 things. We're gonna streak, streak, streak. We're gonna practice, practice, practice. We're gonna use sterile technique. Not too difficult, okay? Any questions on that? That's what you're doing today.
Are we gonna need one of each of these as well?
Yes, yep. You'll need to go into the cooler and get your plate, which is right on this level shelf. You'll see all the nutrient auger plates, and then grab one of this and one of this. Okay? Everyone got that? So, what are we doing? Not yet, not yet. I've got you for another 5 minutes, okay? Maybe 10. What are we doing in here? What are we doing with all this streaking? Okay. On your homemade auger plate, you're simply practicing before we truly address the next year. So, we covered differential and selective media in lecture. We covered differential and selective media in lecture. And I'm as dumb as a rock, so I have a hard time grasping things. So here's how Coach Gray thinks about it, all right? Selective is big. We are selecting for gram positives. We are selecting for gram negatives. We are selecting for coliforms, okay? So selective is bigger than differential. So What's the selective media? Both of these are selective media. One's going to select for gram positives, and one's going to select for gram negatives. Your mannitol salt is going to select for gram positives. Your EMB is going to select for gram negatives. Well, that doesn't help me a whole heck of a lot because there's a ton of gram positives and a ton of gram negatives, right? So that's where the differentiation comes in. Differential, more specific. On the mannitol salt, if colonies form, I got gram positive. If colonies form, and typically on this, we get two types of colonies. We'll get staph aureus, because that's what we see a lot in abscesses and boils and pyodermas and stuff like that. And staph aureus is the problem maker there, okay? And then we have staph epidermidis, which kind of lives on us with the staph aureus, keeps the staph aureus in check, but lives on our body mostly, okay? So, yes, if I have an abscess or pyoderma or I'm culturing something like that, I'll plate it out straight for isolation on this. And if we see little colonies, probably staph, because staph's about the only bacteria that lives in, what's the salt concentration on our skin? Almost 7.5%. So when you sweat, you're putting salt out there, that's making you a tougher barrier to penetrate. And I always I wipe down the machines and the bars when I'm done lifting. And I always thought to myself, why should I do that? Because I just put a ton of salt on that seat. There's probably no one that's going to grow except staph. But we wipe it down anyway. So staph grows in salt. Not many other bacteria grow in salt. So then that's the selection. You're not going to get gram negatives on this. You'll get gram positives only, primarily staph, and there's two staph that we need to worry about. The normal flora that doesn't cause problems, epidermidis, and the aureus, staph aureus, that causes epidermidis, pustules, abscesses, and the likes, okay? So then we want to differentiate. Good Lord, puberty again. What's up, my man? Okay, so if the colonies turn yellow on this, and this is all in your little handout you have, I'm just kind of trying to summarize it for you. If it turns yellow, you've got the human pathogen Staph aureus. If it just stays whitish, reddish, pinkish, something like that, it's Staph epidermidis. Well, why does Staph aureus go yellow to differentiate between all the other staph? It ferments the mannitol in here. Mannitol is a sugar, okay? It's not a sugar you want to put over your Wheaties in the morning, but it's a sugar. And Staph aureus is the only one that really ferments that, and that turns the area around the colony yellowish acid when we ferment. Epidermidis doesn't do that. It doesn't give a crap about mannitol, okay? It just grows on this because it's got salt. So that's what selective media is. Selective is big. And we're selecting primarily usually for gram positives and gram negatives. Well, after we get that, then we start to look at the colonies and whether, they have some special characteristics. That's the differential aspect of a media. And a media can be both. And it is both. Here. And that's what this lab is all about. Using differential and selective media to determine, do I have Staph aureus? Well, I've kind of already given you the answer to that. I told you that's in the mixture. There's no Staph epidermidis in there. So you're going to get yellow. You can get yellow around the colonies because Staph aureus will take mannitol and it will ferment. None of the other staphs do that. That's the differential part. A little bit more specific. We're kind of differentiating between the species of staph. Does that make sense to you guys? Seriously, if you don't get that, it will really be confusing. So selective, big picture, positive, negative. Differential, more specific, species, okay? And epidermidis doesn't cause a clot. You put it up to any one of you, anywhere on your body, and you take a swab, and I can get pretty much epidermidis all the time. When we start to get staph aureus, that can be a normal flora, but in great numbers, causes problems, pyoderma, pustules, boils, abscesses, stuff like that. Cool with that? Okay, so the other media is called EMB. It's selective, and it's differential. So if it's selective, what are we selecting between? Would you guess? Because I haven't told you. What? Good job, good job. We're once again selecting between gram-positives and negatives. The dyes that are in this EMB media, which stands for E is eosin Y, that's a dye. MB, methylene blue, you've used that in your staining, okay? Are two dyes that pretty much inhibit all gram-positives. Gram-positives can't grow it, but gram-negatives love to grow it, right? Now, when we use the EMB, you guys don't use this, CMH anymore. What do you guys use?
We use the McConkey order.
Okay, McConkey, yep, it dealt with that. Probably a little bit more specific, a little bit easier to use.
It also, it's selective, but also differentiates between lactose fermenters.
Right, yeah, lactose and not. Okay, so we're just getting gram-negatives here. And then, which pretty much is gonna be coliforms, depending on where you got your sample. So coliforms, what does that mean? What does coliform mean?
Is it a cluster or something?
What's that?
Is it like a cluster or something?
Well, it's a collection of all the bacteria that live in your guts. We call those coliforms. Oh my God, how many species and genuses are there? Probably thousands. No, I know thousands, because the stuff that I take, that Gundry diet that I'm on, you read the list and it's got like, and there's 100 subspecies of this, and there's thousands of different types. So this is gonna go select, get rid of the gram-positives, coliforms. Of the coliforms, the Enterobacteriaceae, I didn't say that right, but there's a lot that we don't care about. They just don't cause problems, they're not strong enough to do anything, but E. coli causes problems, especially if it gets in the water supply. So we want to know if we got E. coli or not. Well, I kind of already given it away because I told you that one of the bugs in the mixture was E. coli, so we should see E. coli. Here's how you differentiate. Okay, so we selected for gram-negatives, specifically coliforms. Understand that? Selected. This is also differential because E. coli are going to show up as little shiny, metallic looking blue-black colonies. It's really cool looking, okay? And everything else is pretty much blank. And the reason they turn blue-black like that is because they ferment eosin, EMB media, okay? Eosin is the E, and they ferment methylene blue. They ferment the dyes. And that causes, and E. coli is the only one that can do that, pretty much. And it causes that blue-black, while any other colony is just gonna kind of be a cream colored, maybe a little reddish tinge to it, pinkish tinge, something like that. But that's what you're looking for when you use this media. You're looking for that metallic blue-black. Any questions on that? So that's, you know, being dumb as a rock. That's how Coach Gray thinks of selective versus differential. Selective, big picture, and positive and negatives for the most part, and then we're going to differentiate between things that cause a problem, like Staph aureus, versus don't cause a problem, typically Staph epidermidis, or E. coli versus all the other thousands of gram-negative bacteria that we have living in our guts. Okay? Any questions on that? So, coming back full circle to what you're gonna do. You're gonna grab your homemade auger plate, streak for isolation using sterile technique. You're gonna get a mannitol salt plate. You're gonna streak for isolation using sterile technique. You're gonna get an EMB plate, and you're gonna streak for isolation.
With sterile technique, so.
With sterile technique, thank you. 72-year-old brain. Any questions on that?
Can we use these or do we not because they have to be plain?
Yeah, you can use, you can't use, you can, I've got sterile ones back there. If you guys like wanna try the plastic, but it's a one-use thing. You can't use it again, but you know, you take it out and pack it sterile, you can use it. And I've got a ton of them back there. If you guys want that, I'll grab mine here and put them right there. You can try that, because you guys have been using the titanium.
We
Okay.
only have 2 for.
Yeah, okay, and you know, there's just one mixture tube per, island, 6 islands, so if you guys, I'll just bring those out and, set those out there. Are there any questions? All right, so I'll finish up in saying, if you can answer these questions here. You'll do fine on the lab exam. And pretty much this
Is the lab
just
exam next
deals
week?
with selective versus differential, big picture versus small, okay? And the 2 specific media, it's the mannitol salt that's restricting for selective, and it's the mannitol that gets fermented by the staph, that we know it's Staph aureus, because the other staph don't do it. And with the EMB, we know this is selecting for gram-negative coliforms, and of the coliforms, only E. coli ferments these 2 dyes in here, E is in white, and B methylene blue, and turns metallic blue-black. So the last thing, and then I'll let you go, is the results need to be interpreted in about 2 to 3 days. Let's see, today's Thursday, Friday, Saturday, Sunday. So we'll all meet here Sunday. They didn't even yell at me, they're just laughing. Okay. Okay, so really the results need to be looked at, the plates need to be looked at on Monday or Tuesday. Are you guys here on Monday or Tuesday?
You're making arrangements for that, Coach?
I think what I'm gonna, the wife gets so mad at me when I do this. I think I'm just gonna unlock the door, and you guys can come in and take a look, and please don't, she'll castrate me. If she finds out, so, you know, she doesn't like things unlocked, okay, so she thinks we should. Yeah, she's got a point, but you guys are such a phenomenal group. I mean, you guys are superstars. So I will unlock that door on Monday and Tuesday early. You guys are here in the morning? Okay, or afternoon, okay? And here's another way you can do it. If you can get a good picture on your phone, and just text it to the other members. Okay, I'll let you guys decide whether you want to do that or whether you want to come in and take a quick look at it. Does that make sense to everyone? So that's got to be done Monday or Tuesday. Any questions? All right, so I'm on the ringer because I hope I've got bacillus in the spore state, because it's going to be like impossible to isolate if it spreads, because bacillus tends to spread balloons. It's hard to get isolated. So that's what you need to do. There's 2/3 of the plate you need, the other third is back. I'll get these one-use loops up here for you guys, and
Before you get started, I just have one question. And this is a really stupid question, and you guys probably know this, should know this, I know you know this. But when you're streaking these plates, this was a homework question a couple of weeks ago whenever I
I
was
think it's 1 per grade.
grading papers.
I think we do
noticed
1
there was
per.
some misunderstanding maybe of the question.
Sure.
But when you're streaking
You
these
just said he'd
plates,
deliver them around.
when is bacteria being introduced to the plate?
Initially, the first streak.
Right. So you open this, you're doing sterile technique as much as you can, but whenever you're introducing bacteria for growth, it is your initial streak, whenever you have you're essentially inoculating with your culture, right? You take your loop, you sterilely go into the culture, you're out, and then you swab, and then you streak.
And then you sterilize between each other
Right.
streak.
And so you're not reintroducing bacteria after you've sterilized them on your 2nd streak, and your 3rd streak, and your 4th streak. You're not reintroducing bacteria, you're taking that bacteria that
Same
was
bacteria.
initially inoculated the very first time, and you're spreading it thinner and thinner across this slide, right? And so bacteria is being, or your, whatever microbe you're trying to grow, bacteria for what we're doing here, is introduced specifically, you are introducing it on that initial swab, right? When that I'm sure that question was asking, I know that question, the way I interpreted it, was asking for that. It wasn't asking like, well, whenever I took this off, there was some bacteria that kind of fell in there, or whenever I did this, or it must have did it whenever this happened. You guys are introducing bacteria because we are purposefully streaking for isolation. We know that there's bacteria, we're just trying to grow specific bacteria or isolate what bacteria we're looking for. So I just wanted to make sure that kind of the germ theory was kind of understood that we are introducing.
And I wanna add one more thing, Coach, about the mannitol salt. So, mannitol, like I said, is a sugar, and there's salt in there, but those are both clear. It's on a, you know, what that pink part is a pH indicator. So when it ferments the mannitol, it creates an acid. The acid then turns the pink to yellow because of the pH indicator. So that's how it's, you know, more differential, is the pH indicator. And the fermentation of the mannitol turning into an acid. That's all I got.
That's a good point. And all this is in that layout that you read before you came to the lab, right?
Yes.
Okay, all right. So review it, because that's what you're gonna be responsible for.
I don't know, they read for it.
Yep, each one of you will do 3 plates. Each one of you does 3 plates.